My name is Dr. David Feliciano. I'm an assistant professor at Clemson University. My laboratory is interested in brain development, and in particular, we're interested in development of the forebrain and specific regions within the forebrain, including the development of the cerebral cortex. In the laboratory, we study a couple of different disorders, but one of them is what's called a monogenic disorder or single gene disorder called tuberous sclerosis complex. And so those children have epilepsy, autism, intellectual delay. This particular manuscript is focused on the development of the neonatal brain. We're interested in these hollow cavities that are sort of the remnants of embryonic development, but they persist throughout our entire lives. These cavities are fluid-filled. They contain cerebrospinal fluid, and historically cerebrospinal fluid has been seen as a sort of passive liquid that has functioned sort of as a cushion. The holes in the cavity in the CSF is maintained throughout our entire life. One of the questions that we had was whether the CSF really is as passive as it has historically been viewed. Previous reports from our laboratory and from my postdoctoral research demonstrated that there are small particles that are found within the cerebrospinal fluid. And these particles are membranous particles, so they have a lipid membrane, and they kind of function like envelopes. And these envelopes are filled with a letter containing information in the form of microRNAs or small RNAs. There is a question, however, about what the sources of those particles are, what the contents really is, what the encoded information means, and what the recipient cells, if any, are. So this particular manuscript has demonstrated that along the outsides of these cavities that are filled with cerebrospinal fluid, called the subventricular zone, there are neural stem cells, and these neural stem cells release these particles, these extracellular vesicles. What we did was we identified the types of the message inside of these extracellular vesicles, inside of these envelopes. And we demonstrated the envelopes were transferred to immune cells. Now, the developmental origins of these immune cells is quite different than most of the other cells within the brain. The surprising finding for us was not only that the envelopes or these extracellular vesicles were being passaged to the microglia, but also that the microglia seemed to read the envelopes. They read the message. They changed their physiological state. So the immune cells have many important functions in the brain, and they arise within the subventricular zone during neonatal development. And so what we identified was that by changing some of the content or even the numbers of these extracellular vesicles, this could cause the microglia to infiltrate into the subventricular zone at different times. So the hope is, from understanding this neuroimmune axis, we can identify whether there are events that may perturb the axis and turn it too high in one way, so there are too many of these neuroimmune cells, these microglia, or perhaps too low. The other interesting thing is that the results from this study would allow us to perhaps suggest that we could engineer these extracellular vesicles with different types of messages. And the importance of that is that you can take these messages into a diseased brain and you could transplant them. Thereby, you can educate the immune cells within the brain and perhaps even work on rewiring the entire system, or in times of perhaps traumatic brain injury or a viral infection, you might be able to somehow coax the microglia into the right physiological state to help out.